Welcome, everybody. Good morning, good afternoon, or good evening, wherever you guys are joining from. Thank you for being here. I'd like to introduce myself. My name is Aline, the VP of Product at Countable Labs, and I'm very excited to welcome you to today's webinar. From months to days, finally, a low variability analytical assays that work the first time. While we're getting started, we'd love to know, what PCR techniques are you using right now? There should be a poll that has popped up on your screen. It helps us tailor our presentation and our Q&A session so that we can address your most pressing concerns. So also, just to give you a few quick notes before we begin, this session is being recorded, so it will be available on demand. You guys will receive a link by email after the webinar. Also, you guys have this Q&A box at your disposal. You can use it anytime to send in your questions. We'll cover as many as we can during the live Q&A in the end. And obviously, if we miss anything, we'll follow up with you directly. And also, there's a lot of technical guides, blogs, validation data, and more information. Please just go ahead to our countablelabs.com resource center for more of that information. So today, the topic is all about unlocking reliable, fast quantitation for cell and gene therapy workflows. Digital PCR promised absolute quantification, but the reality has been more complicated. Because of Poisson statistics, there's a lot of estimates, assay variability, and very slow assay development. And as a result, many teams still rely on qPCR for convenience, right? Even though the data falls short for them. And for countable PCR, we eliminate that compromise. We deliver digital level performance with a qPCR-like workflow, no optimization, no specialized instrument, and no steep learning curve. Okay, I'm looking at the poll right now. I'm seeing quite a few qPCR users and some digital PCR users, and some are not currently using it at all. So it's a very good mix of crowd today. Okay, great. I think thank you for your results. Here, I'd like to walk us through how it works and how it's getting used in cell and gene therapy workflows like vector copy number, viral titer, and even genome integrity. So today we have Dr. Stephanie Call, Senior Staff Product Manager here at Countable. So Steph, over to you.
Thanks, Aline. Thank you all for joining us today. As Aline said, I'm going to talk about how to use Countable PCR to get low variability cell and gene therapy experiments done quickly and efficiently with great results. So as a product manager, I spend a lot of time talking to users and potential users and just hearing about their frustrations. And in the cell and gene therapy space, especially, what I always hear about is that everyone's just really frustrated about how long it takes to get anything to the clinic or even preclinical trials. So from proof of concept to the clinic, it takes a really long time to collect all the information needed to move your potential therapy through the pipeline. And those months are costly, they add delays, they prevent you from knowing whether a drug is a good candidate or not long-term in the clinic. So what slows down those pipelines? We're here to talk about PCR specifically. And obviously, as our poll kind of indicated, there's two main options that people are using right now, and that is quantitative or real-time PCR and then digital PCR. qPCR, it's fast, it's cheap, it's familiar. Pretty much everybody's done some sort of qPCR experiment, but it's really prohibitive for multiplexing, and the data is not truly quantitative in an absolute sense. So the relative quantification doesn't align with the FDA expectations or other regulatory guidelines. So the idea is to move to digital to overcome these regulatory guidelines. But that's really complex, it's slow to optimize, and it takes a long time to kind of validate those answers because there's a lot of subjectivity involved. And they're just not versatile enough to support the CGT critical quality attributes that you're trying to build up. So I want to dive a little bit further into why digital PCR specifically falls short for a lot of researchers in this field. So DPCR It promises absolute quantification, but the reality is um, there are only about 20,000 partitions, and each of those partitions must contain only one target molecule. So as soon as you start multiplexing, you get inefficient amplification of the different target sequences. And it's really easy to kind of saturate those partitions. So you have to do a lot of dilutions down, which introduces more kind of user error from pipetting differences. And then you also end up with what's called range. So in this example here in kind of the middle of the slide, we see a 2-plex dPCR assay, and the drag on the blue line and the drag on the green line, that's the range from that assay. And then the orange signal is supposed to represent uh, where how many partitions contain both signals. And all put together, all of that rain, you have to threshold around that and it becomes really subjective. So where it takes months is optimizing your assay so that you don't see that rain or you dramatically lessen the rain so that you feel more confident about where you're calling single present, and then the double present. So in order to do that, the reason it takes so long is that you're balancing your probe concentrations. You have to really optimize those cycle conditions, and you have to find that best threshold to reduce variability. But again, that's a little subjective, which is not great when it comes to those regulatory guidelines. And all of that just slows down your optimization process. And going back to how that fits into the bigger picture, when you're working in the genomics, biomarker discovery, early R&D, however you kind of classify that work, you're doing titer quantification, you're checking on the expression and efficacy. But as you move into analytical development in QC/IND, that's where you need that really like strict quantification in order to satisfy the regulatory guidelines. So that's where the handoff between qPCR and dPCR comes in. And even though qPCR lets you scan a lot of candidates or options really quickly, it doesn't really, again, satisfy what those regulatory agencies want to see. So then you have to kind of translate that qPCR experiment into dPCR, and you run into all those aforementioned problems. So to alleviate these concerns, Eileen and the rest of our team helped discover and develop Countable PCR. It delivers the reliable answers you need, and it does so the first time. So whether you're looking to improve on your qPCR or if digital PCR is frustrating to you, Countable PCR really kind of offers the best of both worlds there without sacrificing any data quality. It lets you use one platform to do all of the work that you would always do in qPCR, as well as what you either want to or have already moved into digital PCR. And it allows you to develop low variability assays really, really quickly. You can get stuff off the ground in a couple days that would normally take weeks and even months. And it's also really friendly for the regulatory agency aspect and GMP requirements because it automates your analysis and it gives objective readouts of the quality of your data. So I'm going to talk about all of that. But first, I want to explain what Countable PCR is a little bit more specifically, and also kind of walk through what makes it so unique and what the workflow looks like to set up an assay in Countable PCR. The key crux to why Countable PCR is just so much more powerful than what other tools offer is that it allows you to make 30 million compartments. We use the term compartments rather than partitions, but if you are familiar with digital PCR, it's similar in nature to a partition, but we call them compartments because we're really kind of locking down and truly isolating single molecule targets for amplification. So let's look at the workflow because I think that helps explain a little bit about how exactly we do that. You see here in this picture, this is a spin column and a PCR tube. This is all you need to get started with Countable PCR. Your setup will be taking that spin column, placing it into the tube strip, and in there you load your probes, primer, and template, plus all the ingredients you need to do amplification. And then you also load our special reagents for matrix creation. And you move that into a benchtop centrifuge, spin it for 20 minutes, and that's going to generate the matrix. Now, the matrix is clear. It is occurring in 3D in the bottom of that PCR tube. And it's basically like a gel. So it's semi-rigid. There's not stuff moving around within it. And as you can see from kind of the zoom in picture that we have there, each single target really gets locked into this like picoliter sized compartment. So it's truly isolated and amplification can occur unbiased. So you move the tube into a benchtop thermocycler, and whether your targets are rare or abundant or somewhere in between, all of your targets are going to amplify super cleanly without crosstalk, without amplification bias, because they are so isolated and suspended in the single molecule three-dimensional space. So you don't have to worry about the interference that occurs, for example, in the digital PCR example that I showed. And then that lets us just count directly exactly what's there. In our countable system, there is a light sheet imaging. It moves across the tube like this, and it scans hundreds of light sheets in 40 seconds in order to literally build in 3 dimensions which compartments are giving signal for a given probe channel. And so what does that look like? I have a nice little video that makes it, I think, a little bit clearer. So here again, you're going to have a tube and your light sheet is moving this way. So let's take a look at what that looks like. You can see the counts counting up as we move our way through the tube. And what we're doing is counting exactly how many signals we get in each channel. So each channel represents a different target that we've done. So this is a 4-plex experiment. And you can see there's a pretty wide range of the number of counts in that example. But since it's just counting directly, it doesn't matter. You can look at rare and abundant targets at the same time. And this is a true count. There's none of the Poisson correction required for digital PCR, and there's none of that subjective thresholding with the CT values for qPCR. And again, the counts are automatic, which is really GMP friendly because the computer algorithm is figuring it out for you and giving you the exact same thresholding, the exact same calculation every time. And it images 96 samples in one run. And it does so without sample loss. So it is scanning your whole tube. So you can put a lot of DNA in there to analyze. So if you have, for biodistribution experiments, if you have like really low concentration DNA, you can load more of it in order to pick out the rare biodistribution events more easily. So let's kind of compare that side by side to how it works in dPCR. So going back to the compartments that occur in digital PCR, when you've got 20,000 partitions, you're really struggling with input limitations and just general inefficiencies that occur. So you waste a lot of time optimizing those reactions, as I described before. You get that data with all the rain in it. And one of the problems too is that once you get to higher concentrations of target, you're going to have a lot more variability because the Poisson distribution kind of breaks down once you get above about 100,000 to 200,000 counts, depending on like which specific system you're using. But with Countable, by doing that imaging, we pretty much completely eliminate all of those issues. You're simply counting the signal from the single molecules. It's a lot faster and more reproducible because there's no concerns about dual occupancy, and you get the answers in a lot less time. Plus, again, because everything is really isolated in fixed space, you don't have to worry about the amplification bias that starts to show up when you're trying to do 2-plex and above experiments. So let's show just what that dynamic range means. In qPCR, you have a pretty wide dynamic range to work with, but it's super dependent on having really good standard curves in order to accurately calculate what the concentration of your target is in a sample. So in this example I show here, if you don't use the standard curves, you really lose the accuracy of qPCR. And those standard curves, it actually secretly increase the amount of labor and cost that goes into a qPCR experiment. In digital PCR, again, we talked about the Poisson distribution a bit, but it really breaks down the maximum number of targets that you can count in your sample. And so that really limits you in how you can design your experiments, not to mention all of the other problems that occur when it comes to multiplexing. So now with Countable PCR, we are accurately and unbiasedly measuring across a 10 to the 6th log dynamic range. And we're getting accurate counts per 50 microliters to up to 1.2 million input molecules. And that's all in one experiment. And again, you can load 1 million or you can load 10, and you can load them in the same sample and see all of it. So it's giving you really precise, really linear counts. All right, now that we've seen a little bit more about how Countable PCR works, I want to talk about one more thing that Countable Labs offers, and then kind of get into how and why we can really dramatically reduce the assay optimization time for cell and gene therapy workflows. So we also offer from Countable Labs the universal multiplexing. And the concept here is that we have qPCR simplicity when it comes to kind of probe additives, but you get the true quantitation that you hope for from digital PCR. It's as easy to design experiments for this as really doing like a cyber green experiment, but you're getting all the multiplexing benefits that allows you to compare targets more directly and create more creative experiments, basically, which I will show some examples of later. You don't have to use universal multiplexing. TaqMan probes do work in the Countable PCR system, but we developed universal multiplexing because TaqMan and hydrolysis probes are just quite expensive, and universal multiplexing super decreases the amount of money and time you will spend optimizing multiple multiplex experiments. So briefly, how that works is we have universal adapters for 4 different probe colors. And it goes on your forward primer as a leader sequence, basically, to whatever the rest of your forward primer sequence is. You buy a typical reverse primer. And now as that amplifies, that leader sequence gets tagged onto your amplification product. And the probes that are floating around, they bind only with that leader sequence. So what happens is when it binds, the quencher of the probe kind of releases it and the probe gives signal. So you can see it's pretty straightforward technology, but it allows you to do this hybridization-based probe that ends up costing a lot less because you're just buying standard oligos in order to design this experiment. And just to really drive home, Countable PCR doesn't just give you a lot of flexibility with experiment design, but it actually just reduces cost of like labor and materials for everything. You're getting absolute quantification with it, and you're getting the power of multiplexing, but it is as easy to set up an experiment as your standard qPCR type experiment. But again, you're just getting that quantification power. We have a CLIA lab collaborator who has given us some specific information about side-by-side comparison with dPCR as well. So this is a CLIA lab collaborator who's looking for 0.03 mutant allele frequency in their tests that they offer for patients. In order to do that in dPCR, they have to run 3 wells with the patient sample. And given the number of patients they have per month, that ends up costing them about $26,000 per month. With Countable PCR, again, because we can tolerate a really high volume input and it doesn't matter, it doesn't impact the counting because we don't have that dynamic range limitation, they can consolidate that test into one tube and bring their monthly cost down by about 45%. And that is just one test on a really extensive menu of tests that they offer for patients. And speaking of collaborators, we have another early access customer who is in the cell and gene therapy space. And they've specifically called out the fact that they have basically no more use for ddPCR unless they are trying to validate old runs. So I think this shows just how powerful Countable PCR is, and what a game changer it really is for people in the cell and gene therapy space. So I've talked a lot about, the savings and how it's so great, but let's show you some real examples of how Countable PCR works, and why it's so great. So first we have an example from our collaborator at Carigent, and they've developed new titer assays using Countable PCR. You can see the replicate data of their titer measurement on the left here. Again, because of that broad dynamic range, they don't have to dilute down as much. And you can see, particularly for dilution 1 and 2, that's well above the range that most digital PCR machines can accurately quantify. When that gets back calculated into the actual titer, you can see on the right comparisons they did across a couple different batches. And actually, they were interested as well in moving into universal multiplexing in order to decrease that cost. And what they found is that TaqMan and universal multiplexing perform exactly the same in countable PCR. So you see really good reproduction between the two probe strategies. And then as well as you can see here, they got 1 to 3% CVs on their titer measurements, which again, compared to what I've heard from a lot of people in this space, what they get for even digital PCR is a dramatic improvement. I have heard people are tolerating up to 20% CV error on doing digital PCR titer, and here we're down to 1 to 3% variability. Another example from Carrigen, they've actually found countable PCR so easy and straightforward to use that they've turned to using it as a quick process optimization tool. So what you see here is they have an integration target on a vector, and they are looking to see when their cell line is stably expressing the integration target. So they set up a multiplex experiment. They test their cells once a week, and this has really helped them save weeks on their cell culture process because they have a really easy way to test VCM. Next, I want to talk a little bit more about that titer process. So this is some in-house examples we generated. And I think it really shows the low variability that you get from titer assays with countable PCR. So in this example, we again use multiplexing. And what we did here was design amplification targets for the CMV enhancer, the transgene, which was GFP in this example, and then the poly A tail. And we did titer dilutions for all of it and did the calculation off all of them. And what you can see here is two things. One, no matter which target we use, there's really good linearity and agreement for all of them, and really low error rates when you back calculate the final titer. And then the advantage here is that you are really targeting your actual transgene, which is what you care about when you are designing your gene therapy vectors. And it's obviously quite cost prohibitive to do new probes for every new transgene you're working on. But here, because universal multiplexing is not really cost prohibitive, it's really easy to design hyper-specific assays that really interrogate what you really want to know, which is my transgene there and how much of it is there? So this lets us just dilute less sample. Again, we're still capturing accurate titer measurements. And again, you can do 4 different regions of interest if you want from your vector and put it in one tube, and really no optimization. This took our R&D team a couple days to get. Now, using that same vector, we inserted a restriction digestion site in between the CMV enhancer and the GFP transgene. And then what we wanted to do was kind of mix the undigested and digested at different ratios in order to test linkage. So first, what that looks like a little bit, this is a representative light sheet from each of those channels, so it's kind of layered on top of each other. And what happens is, because we're building that sort of map in 3D, the computer algorithm that does the linkage analysis can actually map exactly which ones are oriented in exactly the same compartment space. So what you get is this composite map, and that's telling you what the different signals are. So you've got the GFP poly A partial, you've got the CMV enhancer partial, and you've got the complete genome. And so again, we mix those in different ratios, and what we found is we get the genome integrity ratio quantified with really high precision. You can see the dilution factor, that's complete genome over the amount of partial genome added. And it reflects really well what the expected values would have been. Again, the data is generated automatically. You just get counts out of this. There's no pause on statistics or modeling. And there's no concerns if you were trying to do this with NGS, you're not skewing your results with library prep bias. So one last kind of interesting experiment we did in Countable PCR is we wanted to interrogate whether you can replace long-read NGS with long-read PCR. So this is about really knowing if your entire vector is present and accounted for in your experiments. So we designed sort of a walking amplicon from the CMV enhancer all the way down to the poly A tail. And that makes the max amplicon length about 2,000 base pairs. We ran it in countable PCR, and what we found, again, we did kind of TACMAN and universal multiplexing just to validate the performance is the same for both. And you get really good agreement for complete genome when you use this method. Secretly behind the scenes, our R&D team has also gone up to 5,000 base pairs with very similar performance. And it just lets you ditch this almost this technical limitation of 150 base pairs that both qPCR and dPCR kind of stick you with. So you can get more information about a longer piece of your gene of interest. And this lets you be a little bit more direct about quantifying your genome integrity. And it's also great for quantifying residual DNA. And again, you don't have to do NGS, so you're saving a lot of time and cost on library prep, the sequencing and analysis time. So one last thing I kind of want to touch on is just really hitting home how we're helpful in that GMP and regulatory environment. Because countable PCR is directly counting, it's going to tell you exactly how many are there. You're not doing any mathematical wizardry or subjective thresholding in order to come up with that number. So first of all, for each amplification event, we offer an ID score. So this is great for GMP because it doesn't just tell you, hey, this is the number you got. It tells you, hey, this is the number I got, and this is how confident I feel in this number that I'm reporting. So again, when you go back to these regulatory agencies, or even if you're just working across cross-functional teams, you can go back and say, hey, this ID score showed that this experiment worked really well, and we are really confident in the results. And then again, the software outputs direct counts. This is an exact result from an experiment. You can see we went from about 800,000 target counts down to about 100,000, all in the same experiment. And it just tells you exactly what number is there. So no subjectivity here. It's just exact counts. As we wrap up, I want to share just a couple other kind of customer testimonial pieces. First, we have a collaborator, Rick Tarleton at University of Georgia. They use their instrument to investigate Chagas disease. And what he finds really powerful for this is the million-fold range in a single reaction. It really helps them get precise sensitive detection of their parasite. And then, of course, our friends at Caragent, they are really excited about replacing NGS for their work. NGS is costly and expensive, and being able to move more of that information into Assays Design and Countable PCR is really great for the services they're trying to provide for their own clients. So moving on, last, I just want to kind of share what Countable Labs has on offer. So we have Assay Design Services. There's a couple different tiers. I'll talk about those a little bit more in a second. But then we have all of the supplies, materials, consumables you need in order to set up your multiplexing and generate the matrix that's so critical for this precise counting. And then, of course, the system and the software for actually doing the counts and getting your data. To touch on the Assay Design Services a little more closely, we have a couple different levels, but if, for example, you are interested in getting started with Countable PCR, but aren't ready to buy your own instrument yet or need time to do that, we offer full project, which is white glove Countable Labs partnership. We will really work hand in hand with you to fully design your experiments from start to finish. Make sure that they're kind of the most efficient and best experiments that you want to do, and give you all of the data output that you need in order to know everything you need to know about that experiment. So with that, I hope we've all learned how Countable PCR is helpful in the cell and gene therapy space. I see Eileen's back, so I'm sure there have been questions while I've been talking, so happy to answer what we can get through.
Okay, thank you, Steph. That was a great walkthrough of how Countable PCR simplifies and speeds up quantification workflows in the cell and gene therapy space. So now, well, let's go on and move into the Q&A portion of the session. If you haven't done so already, feel free to drop your questions in the Q&A box. We'll take as many questions as we can, given the time we have left. So we already have a few coming in, so I'll jump right into it. Okay, so Steph, what's the minimum and maximum amount of DNA you can load per reaction?
Great question. So, like I said, we're analyzing the whole tube. There's actually a lot of room for volume-wise DNA to add, so you can put up to 35 microliters of your target DNA. And then if you're working with a sample that's got really dilute target, you know there's not going to be a lot of it, you can load up to 1 microgram of DNA as well. And then on the low end, we don't recommend much lower than about, what is it, about 1 nanogram is about the lowest. So if you've got like a very concentrated sample. And then of course, on the volume side, it's whatever you dilute it out to. So, on the low side, it's kind of up to you.
Okay, thank you. And the next question seems to be for probably a vector copy number because it talks about gDNA. Should it be like pre-digested before loading into the countable instrument?
Yeah, performance is generally best if it's digested. Eileen, do you have you've done more of these experiments than I do, so I don't know if there's
Maybe
any.
I should, yeah, you're right. I mean, it really depends. We've done both
Okay.
undigested and digested gDNA, typically like extracted gDNA from any commercially available kits, would work really well in our system. Depending on your answers, we have folks who like to pre-digest it because of secondary structures that would lead to undercounting, or people who want to look at concatemers right next to each other. Then they either want to cut it or not cut it. So it depends on your application where you can do both. Okay. How many colors or channels does your system have?
Yeah, so we have 4 channels right now. So blue, green, orange, and red.
There's a question I think about probably about variability. So what is a typical percent CV you would expect to see on the countable instrument?
Yeah, I would say on average from most of the experiments that I've seen, like right out of the box with almost no optimization, you can get even lower, but like max 5% CV. And honestly, our techs who are very good at pipetting get like 1 to 3% CV. So even I, who haven't really been at the bench in ages, get really good CVs when I run experiments in countable PCR.
No, yeah, completely. Once you get into the single molecule space with 30 million compartments, you're just counting one molecule at a time. So I want to add that most of the time you're probably measuring the operator's pipetting error. And typically what I've seen is 1 to 3% CV, which is kind of amazing. Okay, thank you for the question. Let me scroll down a bit. I think somebody is asking whether we can detect RNA or like can we use cDNA? It's probably for like lentiviral workflows.
It's a great question. So, yes, you can. We actually have a one-step RT-PCR kit coming out soon that will be usable for this. It works really great. So, we don't have data to share quite yet on it, but yeah, it's super easy to do. And it's actually another example of really being a huge cost and time saving because you're just putting everything in one tube and letting it run rather than having to transfer and do kind of the expensive cDNA generation step separately.
Okay, thank you. Um, let me maybe this is for me since there's a question about can you use cybergreen for target detection? Um, so the answer is no, we don't support cybergreen. We developed what Stephanie was introducing, we developed something called universal multiplexing. which is to me like an evolution of cyber green. Like you're always looking for more than one target. Even for titer, you want to look at different parts of the gene of interest to see if your titer is accurate, right? But there's no low cost way to do so until now. So with that sort of new chemistry, Countable PCR does not support cyber green, but then the cost of getting basically cyber green like counting like assays is actually much more cost effective by moving to universal multiplexing. Cool. Let me see. There's a question about, are the excitation source and emission filter coupled or decoupled? For example, can I excite in the green channel but measure in the orange channel? Great question. I can answer that. So currently it's coupled. So if you have a FAM fluorophore, for example, we excite in blue and then we have a paired emission filter. The system is designed in a way that the fluorophores are very distinct in the emission spectrum. So for example, like if you have FAM and hex, in other PCR platforms, they tend to bleed over to one another, which makes it really hard to discern sometimes whether it's crosstalk or is it a spectral overlap. But our platform is actually a laser-based system. So you can see here on the slide, the blue, green, orange, red is very distinct from one another. But if you're interested, I would like to know what kind of applications you would use it for, and we can chat more about that. A great question. And then another question is, how do you ensure you don't miss a count if two signals are very close to each other?
Yeah, great question. I'm going to go back to the counting video as an example here. So like I sort of described, everything is really in its own individual compartment. And even if something ends up in the compartment next to each other, because we're basically generating the picture in 3 dimensions, so the computer is almost analyzing like the globe that is emitted from the probe. Our computer algorithm is really, really good at picking that out, even if the two kind of dots are really close together. And we actually have a machine learning algorithm that can be incorporated into any assay. So if you are really concerned about something like that, it uses training samples in order to literally learn the difference between the background, like a true signal and a non-true signal, for each experiment. So it just makes it like even more confidence basically in really picking out those separate signals.
Yeah, thank you. That's great. Yeah, like the video explains it all. I think for example, we've done a lot of experiments. Say like if I add like expected counts of 3,000 molecules, I get 3,000 molecules back, or like put 5,000, I get 5,000 back. So it's actually very linear. So just to prove that our ability to discern single molecules is actually really high. Okay, so we're a bit over time. I just want to give like the last question. The last question is, digital PCR is generally more resistant to inhibitors or quote unquote 30 samples compared to qPCR. Would you say countable PCR is similar? Maybe I'll take this one. So just to give everybody an overview, qPCR, right? Typically you amplify everything in a giant soup. So unfortunately, right, if you have very rare things, it's really hard to amplify that very rare thing in a giant pot of soup. So there's a lot of like evidence in the past decade about how digital PCR is able to partition, DNA, where then you basically have, what you want to see becomes more basically easier to amplify. So imagine this, but we have 1,500 times more partitions than digital PCR. So I would say this is actually even better. So that is actually all the questions we can answer today. Thank you again for everybody's participation. There's a lot of questions. And just FYI, we also have a last quick poll on your screen to see if you would like to be contacted to understand more about the technology. If you're interested and want to learn more, there's also countablelabs.com, which we have a lot of technical resources for you to, learn about countable PCR. We have application notes and blogs on like how we explain fundamental differences on the technology and other helpful videos. Thanks again for joining us, guys. We look forward to supporting your next project with faster and more reliable quantification. Thank you very much.
Thanks, everyone.